David Llewellyn, from master student from the Master of Science in Horticultural Science at Dr. Yubin Zheng's lab at the University of Guelph. He has been working in the horticulture sector for the past 20 years and with a broad experience including field vegetables, nursery crops, landscaping, tender fruits, woodland management, and greenhouse production. For the past 2 decades, he has specialized in controlled environment horticultural research, including greenhouse horticulture production, indoor and greenhouse cannabis production, and engineered interior landscaping for improving air quality in occupied spaces. His main goal is to serve the horticulture industry by performing grower relevant research that will improve production of efficiency while reducing the industry's environmental footprint. So we'll now ask Dr. David Llewellyn to start his presentation. I guess it takes a few minutes to switch over. We have some more questions on which I'll now address at the end of our session here. So Dave, please take over.
Okay, good morning, everybody. Can you see my screen?
Yes.
Anybody? Yes, all right, sweet. Okay, so what I'm going to talk about today, hang on just a second here, are some experiments that we have done that use light spectrum to manipulate plant growth and flowering in greenhouses. I'm going to start with a brief overview of LEDs and talk about some potential spectral interactions with plants. Then I'll touch on what we've learned about spectrum in growth chambers. And finally, I'm going to show how we're trying to leverage that knowledge for use in greenhouse environments. So briefly on LEDs, compared to HPS, current LEDs are approximately 50% higher efficiency, or at least they're approaching that, than HPS. They also have much longer operating lifetimes. And for us, what's really interesting is they're adjustable, meaning that they can be dimmed so you can adjust the light intensity. And this can be quite useful in DLI control algorithms, which is something I presented on at a past greenhouse conference. As well, some LED fixtures, you can also adjust the spectral distribution, which makes the LED fixture a pretty powerful tool. Okay, this image shows the relative absorption of some of the major plant photoreceptors to different wavelengths of light, which is on the x-axis. Note in particular, the green line and the light blue line are chlorophylls, and the light red line and the dark red line is phytochrome, which is something we're going to talk about later. And given the complexity of this image, it's quite easy to imagine that targeted spectrum treatments using narrowband LEDs could be used to manipulate how plants sense and thus respond to their lighting environments. So in the indoor environment, this is where we started our research. We have complete control over crop lighting, including what intensity we can use, as well as photoperiod. And then moving on to spectrum, there's all sorts of manipulations that can be done. For example, you can choose specific spectrum recipes, such as looking at the typical magenta colored LED fixtures versus white LEDs. Also, you can manipulate spectrum through time, like over the photoperiod, or change the spectrum between day and night, just like Quming was talking about with his trial with tomatoes. And finally, you can change spectrum throughout the plant's growth and reproductive cycles to cause specific effects. So our lab has walk-in growth chambers that we use to do much of our research. And this allows us to separate lighting treatments into 6 individual compartments while maintaining uniform temperature, relative humidity, and CO2 throughout the whole experiment. Oops. And in the past 3 years, using these chambers, we've actually published over a dozen papers on using light to manipulate crops. And don't read all these now. I've just added them here so you can reference them later. I would like to note that a major focus of this work has been the effects of blue light, particularly on the photoreceptor phytochrome, which I'm now going to describe and quickly summarize some of our growth chamber findings. This gets a little complicated. So phytochrome, some of you I'm sure already know all this, but I always have to walk through it and remind myself. So here we go. Phytochrome is one of the most important photoreceptors in plants, and it's responsible for mediating a broad range of different plant responses, both on plant growth, seed germination, plant morphology, and flowering. And this graphic shows that there's two interconvertible isoforms of phytochrome, and I'm summarizing here, but basically the two forms are PR, called phytochrome red, and PFR. And the absorption of red light causes a conformational shift from PR to PFR. And PFR is considered the active form of phytochrome. However, I find this hard to wrap my head around. Active phytochrome actually inhibits stem extension. So that takes a bit of thinking to keep in mind. Anyway, this conversion from PR to PFR happens relatively quickly. Conversely, absorption of red photons will convert PFR back to PR. This happens relatively slowly, but also importantly, it also happens naturally in the dark. And incidentally, this slow reversion is why day length, or actually night length, is critical to controlling flowering in photoperiodic plants. Removing a lot of the noise from that spectrum graph I showed a couple slides ago, this is the absorbance spectrum of phytochrome, the two isoforms. And note that these two curves overlap quite a lot, particularly in the red and far red regions. And this means that there's normally a balance between the two isoforms in plant tissues. And we call this the phytochrome photostationary state, which is a mouthful, so we shorten it to PPS. Sometimes it's called PPEs, for phytochrome photoequilibrium. Anyway, this term, PPS, estimates the percentage of phytochrome that is present in the PR, the active isoform. The maximum PPS value is about 0.9, meaning 90% of the phytochrome in tissues is in the PFR form. And PPS values that are lower than 0.9 are normally indicative of environments that are higher in far red, such as under vegetated shade or under light sources that have high levels of far red, such as incandescent bulbs. Note that while phytochrome absorbs most strongly in the red and blue regions, the red and far red regions, there's also some activity that goes on in the blue region, which I'm going to highlight here. This implies that spectra with high levels of blue might affect plants similarly to environments that have high levels of far red. And the following slides are going to show that this is indeed the case. I should also note that the relative responses, so sort of the height of these graphs, is much lower in the blue region than in the red region, which actually has ramifications if red light is present in the spectrum. Okay, on to some results. So this was a growth chamber trial where we had monochromatic spectrum treatment. So we were growing microgreens with pure red light or pure blue light, and we were looking at a range of PPFDs from 200 all the way, or 20, sorry, all the way up to 650 micromoles per meter squared per second. You can see that regardless of the spectrum, higher light levels cause plants to grow shorter, and there's more biomass. And this is normal behavior. But what's more interesting is that the blue light treatments made plants grow even taller than red. And this is the expected response based on what I discussed in the previous slide, but it still may surprise some people since it's commonly believed that blue light keeps plants compact. So the following slides are going to summarize trials that looked at spectrum treatments with different PPS values, including pure red, which is about 0.9, pure blue, which is 0.5, mixture of blue and red, mostly blue light contaminated with a little bit of red light. And then that spectrum mixed with increasing amounts of far red light, which causes incremental reductions in the PPS values. And in microgreens, what you'll see is blue light causes plants to stretch. You mix blue and red together alone and plants stay very compact. And then we see this sort of incremental increase in height as the PPS values go down, which is kind of cool. We see the same results in potted flowers and bedding plant seedlings. And even if you carry those through to flowering, you still see the same sort of patterns. Though things do start to get a little more complicated because of photoperiod also starts to become an issue. So some of our plants don't look so awesome. Okay. So to summarize our findings in growth chambers, pure blue light induces some shade avoidance responses in many species, primarily stem elongation, or that's the easiest to see. However, when blue light is mixed, even with a little bit of red, plants grow more normally, more compact, indicating that the red light overpowers the blue light's effect on phytochrome. Further, you add a little bit of far red, and this actually counteracts that effect, and there appears to be sort of a dose response. With mixed spectra, it appears that there's sort of a PPS threshold somewhere around between 0.6 and 0.65, whereby the blue light activity on phytochrome is offset by either other spectra overcoming that, or possibly also the blue light impacting on other photoreceptors that red and far red don't see. For example, cryptochrome and phototropin. Oops. In most cases, the treatments with blue plus a little bit of red plus a little bit of far red resulted in the best combinations of morphology, flowering, and yield. So we use this spectrum combination in a greenhouse trial that I'll talk about shortly. So on to greenhouses, the meat of the talk. So now we're pretty confident about some of the spectrum effects in the indoor environment, and we find these to be really interesting. But what's even more interesting is trying to figure out if and how we can leverage these findings for use in greenhouses, which is a whole different animal. In greenhouses, there's a much higher variability in lighting, both in intensity, in intensity, spectrum, and photoperiod. And these vary throughout the day. They vary through different weather conditions and also throughout the seasons. Here's a natural sunlight spectrum. That's about 500 micromoles per meter squared per second. And during the daytime, it's kind of overwhelming, or the sunlight spectrum is too overwhelming to apply an LED spectrum treatment, such as the one I've just added here, to have an effect. So I've just combined those two together, and the dotted line shows the LED plus sunlight. And what you see is there really isn't that much of a change. It's not a big change in the spectrum. So what this means is, basically, in order to use spectrum in greenhouses, we have to focus on nighttime. Also, end of day, but that's another, twilight periods is kind of has to be treated specially, I would say, and we're not going to talk about that. So, for example, here's an experiment we did, again, in Soul Source, not greenhouse, but it was with the eye to proving a concept and moving into the greenhouse environment. So the objective of this experiment was to do nighttime spectrum treatments, so different than the daytime, to increase the height of microgreens to enable machine harvesting. And this is because manually harvesting microgreens is the single greatest labor cost in microgreen production. So if we can reduce that by doing it with machines, it's a win for everybody. Now, I'm not going to go into detail on what all these spectrums mean, but I've highlighted underneath. Basically, we divided the night into two halves, the first half and the second half, and applied different spectrum treatments over those two halves. And what we found, comparing to the dark controls in mustard and arugula, is that our nighttime spectrum treatments, all of them basically, increased plant height substantially. Focusing on blue, while the height increases were less substantial than when far red was present, the yield impacts were much higher. The plants grown under monochromatic blue at night had much higher yielding quality. So this experiment provided proof of concept for potential to be able to manipulate plant morphology using low intensities during nighttime. It also confirmed that blue light could mimic some of the morphological influences of high far red environments, even when only applied during nighttime. And with this, we became more curious. We wondered then if blue light could also have similar effects on flowering tissues. And we also wondered, might it be possible to actually lengthen the day, thus increasing DLI, while also tricking specifically short day plants to induce or continue flowering in long days, which has ramifications not only on some floriculture crops, but also in cannabis. So our main question is basically, can nighttime spectrum treatments be used in greenhouse production to improve crop morphology and flowering? And to investigate this, we did two greenhouse experiments. First experiment investigated nighttime applications of blue and far red to increase height without affecting yield and quality of greenhouse grown microgreens. So it's kind of a replication of the trial that I just showed you, but we're actually doing it in the greenhouse environment now. The second experiment looked at nighttime applications of pure blue and that blue plus red plus far red spectrum from previous experiments in the growth chamber on both morphology and flowering of plotted floriculture crops. So experiment one, there were 3 treatments, a dark control and 2 nighttime treatments, which were either pure blue or pure far red. And these were applied for the whole nighttime period. This is what the spectra look like, and I know it's small, but you can see the PPS values here, 0.48 and 0.15, are both below that 0.6 sort of threshold that I talked about earlier. Also note, the PPFDs were very low, only 14 micromoles per meter squared per second. And we grew the plants in this experimental greenhouse environment. There were 3 side-by-side adjacent greenhouses where we had 3 benches in each greenhouse. So this allowed us to do those 3 treatments and replicate each one 3 times, all concurrently. I should note that we used bench level blackout curtains to, when the lights were on, to eliminate spectrum contamination between adjacent treatment plots. So here's kind of what the results look like. And on the top is arugula, and down below is mustard. So these are the plants right before harvest. And what you can see is that we had substantial impacts on morphology with our spectrum treatments. However, should note they were lower than the previous indoor experiments that we've done, which was kind of expected. Note that the plants in the blue treatment here were taller, but also more robust looking than either the plants in the dark treatment. This is dark at nighttime, they had a regular daytime, and far red. In particular, far red, you can see in the bottom picture, also increased height, but at the cost of weaker, more spindly looking plants, basically. And in fact, in both species, the yields under the blue light treatments were about 30% higher than the dark treatment. Interestingly, in mustard, blue light also increased leaf area by about 20%, but in arugula, blue light increased leaf thickness also by about 20%. And this kind of demonstrates that there's species-specific responses in some morphological parameters. So based on these results, we think that there are some commercial applications for using targeted spectra treatments for manipulating plant morphology, at least in vegetative plants. So the next question is, what about flowering? So on to potted flowers. In this experiment, we looked at 4 photoperiod commodity groups, looking at chrysanthemums, geraniums, calibrachoa, and gerbera. We use the same setup, basically, slightly different treatments. In this case, the second end of day treatment or nighttime treatment was the blue, red plus far red. Spectra are here, so the PPS now we've raised it to above that 0.6 threshold. These were only applied for 4 hours at a night, which gave us a very low nighttime DLI of only about. So we didn't add much photosynthetic light with these treatments. This trial started in early February during week 5 and ran till late April. Obviously, the average DLI increased over time. And note that at the beginning of the trial, we had a 10-hour natural photo period, and by mid-March, that increased to 12-hour days. Also note that even though we had 10-hour days here, the 2 end-of-day treatments actually increased the day length to 14 hours. Here's the results. Starting with chrysanthemum, which is a short day plant. I'll point out a couple things. So we have 2 graphs here, flowering index and number of flower buds per plant. The flowering index is reference the pictures up above for that. So looking at it, the dark treatment is the black line with the clear diamonds. The blue treatment is blue line, and the other treatment is purple. What you'll see is that the blue plus red plus far red treatment completely inhibited flowering. However, under the blue treatment, even with long photo periods, 14 hours plus, we did see flowering, and this was quite a nice surprise. And while flower induction was delayed for a couple of weeks, by mid-March, around here, the blue treatment actually surpassed the control treatment in terms of number of buds produced. Moving on to calibrachoa, we had almost the reverse effects. It's a long day plant, or facultative long day, so maybe that's not too surprising. In this case, both nighttime spectra treatments had better flowering than the control. However, the blue treatment lagged behind the blue plus red plus far red treatment. And this implies that there are both photoperiod and spectrum effects on flowering calibrachoa. Also, the other 2 plants, we saw no treatment effects on flower. Moving on to vegetative growth, we took a lot of measurements, but I'm only going to discuss a couple of them today. Notice that the nighttime treatments here increased canopy height of chrysanthemum and geranium, but not the other 2 species. Increased height is generally not a desirable outcome for most floriculture commodities. However, on the plus side, the nighttime spectrum treatments generally also increased biomass metrics, and in many cases, far more substantially than could be explained by the minuscule increases in the DLI. That's pretty cool. Moving on to pictures, not as reliable as measured data, but they do emphasize general trends in treatment responses in morphology and flowering. In some cases, nighttime spectrum treatments actually produce higher quality plants, with or without impacts on flowering. I like to focus on chrysanthemum here, which may be the most interesting. Firstly, we definitely showed that you can flower a short day plant under long days if you give it the right spectrum. That's pretty cool. Secondly, looking at this plant, which didn't flower, but if you're growing chrysanthemums for stock plants, for example, these treatments may actually enable you to produce a better quality stock plant for taking vegetative cuttings. Okay, I'm going to wrap up. So we think we've shown there's potential for spectrum treatments to be a useful tool in greenhouse production in some scenarios. We've also shown that only low intensities are required for these to have profound effects on morphology and flowering. And in dimmable fixtures, blue LEDs can be multimodal. They can play dual roles, both as photosynthetic lighting during the day and photomorphogenic lighting at night, thus potentially increasing the value of some LED technologies. I'm not going to go through future research because I think I'm out of time, but you can see these later if you want. I would like to thank and acknowledge our funding support up top, our industry partners who supply plant materials and ideas, and also lighting companies that provided lighting fixtures for this research. And with that, I'm ready to take questions.
Thank you, David. That was a very nice presentation there. And it's interesting to see that you're pretty well supported what Xiuming said in his talk with the blue light, and that's extremely nice to see that. Extremely good. Thank you. Questions? One from Ron says, does a curve seen with a side photo show a too close light source? I'm not sure which talk. I'm assuming that's a talk for yours, Jason.
I didn't understand that question.
Does a curve seen from the side photo show a too close light source? I think it's talking about the light fixtures being too close, causing some sort of change in the curve. I'm not sure.
Yeah, I don't know either what that means. And our light sources are generally not that close to the plants, particularly for the PPFD levels we're using. So there's not generally any sort of heating issues, and we struggle really hard to get good uniformity over our experimental plots.
Okay. I don't see any other questions come up. Here's one. Do you have the plans to do you have plans to do nighttime treatment experiments with cannabis, or can you already make some predictions here?
I would say yes, we do. And we've actually done this already. We've started looking at not nighttime spectrum treatments yet, but photoperiod manipulations on cannabis. And we have all sorts of ideas, and more ideas than time or space to do them. But yeah, that's a definite goal that we're focused on doing more research on.
Okay, because just looking at your changing the flowering ratio with the light spectrum really changes the need for the blackout curtains in the summer. So if you use that to change that component would be very, very good, and cut down the electricity costs and cooling the greenhouses down. So that'll be kind of interesting to see what happens there. And I think that's the questions I have for you right now. As we get more questions, I'll bring them up later on.